What is virus? For more than a century, the virologists have struggled to find an undeniable definition. It becomes a problem when somebody discovers a virus that doesn't fit each time they think they have found a good definition. Thus, the definition has to change. But we can define the virus as an infectious agent that is not a cell, which consists of genetic material in the form of nucleic acid molecule, usually in a protein coat, and is capable of directing its own reproduction and spread inside the host cell. In 1898, Dutch scientists discovered a disease that was identified in tobacco plants, which took the form of mosaic of light and dark green areas on the leaves. This very first virus was named after the host and the symptoms it caused, which is tobacco mosaic. The true nature of bacteriophage and other viruses wasn't clear until the invention of the electron microscope in 1930s. The first image of tobacco mosaic virus was published in 1939 by Helmut Ruska, a German scientist. In 1935, the American scientist Wendell Stanley was able to make crystals of highly purified tobacco mosaic virus. The crystals of tobacco mosaic virus were used by Rosalind Franklin in the 1950s to determine the detailed structure of the virus in a technique called X-ray diffraction. Franklin used this same method to examine the structure of DNA, and her studies were used by James Watson and Francis Crick to reveal the double helix nature of DNA. In order to standardize the way viruses are named, the International Committee for the Taxonomy of Viruses (ICTV) was formed. Their first report was published in 1971 with 290 species of viruses. The ICTV, composed of virologists from around the world, has developed a complex naming scheme that uses Latinized forms of virus names with species, genera, families, and orders. There are 7 classes of virus in David Baltimore's classification scheme. Class 1 viruses have double-stranded DNA genome. Class 2 viruses have single strand of DNA genome. Class 3 viruses have double-stranded RNA genome. Class 4 viruses have a genome of positive sense single-stranded RNA genome. Class 5 viruses have a negative sense single-stranded RNA genome. Class 6 viruses have single-stranded RNA genome that replicated with DNA intermediate. And Class 7 viruses have double-stranded DNA genome that replicates with RNA intermediate. Replication is a process where the biological viruses are formed in the target host cells during the infection cycle. Viruses first need to get into the cell before the replication can take place. The virus continues to infect new hosts through the production of abundant copies of its genome. Each type of virus in the Baltimore's classification scheme uses a different strategy to replicate. Many class 1 viruses, those with double-stranded DNA genomes, use an enzyme called DNA polymerase to replicate their DNA borrowed from their host, even though they typically also produce some of their own proteins that are involved in their replication. While for class 2 viruses, they convert their genomes into double-stranded DNA before replication. This is replicated through a replication mechanism called the rolling circle. Viruses also use a wide variety of methods to get from one host to the next host. There are 2 main types of transmission: horizontal transmission and vertical transmission. Horizontal transmission occurs from one host individual to another, while vertical transmission is a transmission from parent to offspring. Horizontal transmission happens when a new host breathes in virus particles in the air, while most viruses of wild plants, in contrast, are transmitted vertically through the seed. Viruses have an intimate relationship with their hosts. They are completely dependent on cells for every phase of their life cycle. Although we often consider viruses to be pathogenic, they are not necessarily dangerous. Most viruses are probably only obtain what they need from their host without causing any harm, because a virus might not be able to replicate as well in a sick host as in a healthy one, especially if sick hosts are less likely to mingle with other potential hosts. Plants have a very different immune system. Innate responses to viruses are sometimes specific for the virus and the plant host. Some viruses, for example, initiate a plant response that holds the virus in the initially infected cell and prevents it from spreading to other tissues. These are called local lesion responses. Some viruses trigger an innate response that also affects other pathogens, so that the plant is ready to defend itself against other invaders. Plants also have an acquired immune response known as RNA silencing, but they do not appear to have any memory built into this system. Differences between tobacco mosaic virus and COVID-19. Tobacco mosaic virus is a plant virus. For many plant viruses to be transferred from plant to plant, damage to some of the plant cell must occur to allow the virus to enter a new host. Plant viruses can be transmitted by a variety of vectors, through contact with an infected plant cell or by living organisms such as insects. In contrast with COVID-19, the virus spread from person to person in close proximity. Droplets of an infected person's body fluid, such as saliva or mucus, are spread in the air or on surfaces by sneezing or coughing.